Welcome! Now we're going to talk about TDP-43. It's really funny. My husband went to an ALS conference with me, and he's not a scientist, and he was like, Gently, what is this DP4 protein they keep talking about? And I'm like, what are you talking about? He's like, TD. And I was like, oh, TDP-43! It's really important. And a lot of treatments are really focused on this for C9 ORF72, ALS and FTD, but also several other genetic forms. So what does TDP-43 do, and why do we care so much about it? Well, in a separate video, I explained about this loss of function and gain of function hypothesis. But in order for you to understand what happens when we lose TDP-43 function in the nucleus, you need to understand what it does. To understand TDP-43, you need to understand the central dogma of molecular biology. DNA to RNA to protein. Okay, so DNA is the blueprint that you would hold in a safe, and the safe is the nucleus. RNA is an intermediate blueprint that you would take out of the safe that you could use and you can make copies of. That is RNA. Protein are the buildings that you're making in the city that is the cell, in the factory, and it really allows you to do everything that the cell is able to do. And cells, of course, they work together in unison to make tissues, and tissues make organs. And ta-da, you've got a whole human being. But that's for another day. TDP-43 is an alternative splicing factor. And so what that means, and this is kind of mind-boggling, is that DNA doesn't just go to RNA and protein. The RNAs have different versions that can be made after the initial RNA is made to make different proteins. So it's not just a straight line, DNA, RNA, protein. It's DNA, RNA, protein 1 via RNA 1, RNA 2, protein 2, and a bunch of different types that can be made from the same genetic material. And this is a reason why our genome is relatively small for all that it can do. So TDP-43 is so important to making a bunch of buildings in our cell function normally. What happens when TDP-43 isn't working in the cell? We think this is really important to contributing to ALS because there's a lot of other proteins, some that are really, really important, some buildings that are really important in the city of our cell that are not getting made. And when the buildings are not being made and functioning properly in a neuron, well, neurons are pretty flimsy and then they start dying. And so in this way, TDP-43 is very important to both understand how it can contribute to loss of function. This has two implications, and some of our scientists are talking about these. One is biomarkers and one is therapeutics. So not only does TDP-43 function in the nucleus to make different proteins in this parameter, different buildings, but when it's not working in the nucleus, you can actually get really weird buildings that you shouldn't be making. Let's say a neuron should not have an amusement park. It should have a post office and a hospital. Well, obviously it's bad if the post office and hospital isn't made, but if there's an amusement park where the hospital should be, then we can try to detect that amusement park to say that neuron doesn't have TDP-43 inside working. And so if you do that, you're detecting a signal that implies that TDP-43 has loss of function in that cell. And that's what we call a biomarker. And you can develop these to try to predict someone who's going to get disease later. You can use this to test that someone corrected TDP-43 function in a cell. This has a lot of implications for TDP-43. Understanding the loss of function of TDP-43 is also important to be able to understand the therapies that are being used to target it. Some people are working to prevent TDP-43 from creating clumps in the cytosol and ushering it back into the nucleus so it can do its job. That would target both gain of function and loss of function. Some people are trying to replenish the TDP-43 that is being lost and putting a good copy that can't gunk up outside of the nucleus and create and wreak havoc inside the cell. And Phil Wong is talking about that. Thank you for listening, and I hope to be able to catch you up on key principles in molecular biology in future videos. You can reach out to End the Legacy if there's any topics in particular that you want to hear from. And if there's any experts in particular that you would like me to interview, please reach out as well. Bye.